So it's a couple of minutes after 12, so I think we can get started here. So before beginning today, we would like to respectfully acknowledge the island of Newfoundland as the ancestral homelands of the Micmac and Beothuk. We also recognize the Inuit of Nunatavut and Nunatuckavut, and the Innu of Nitassinan and their ancestors as the original people of Labrador. We strive for respectful and equitable partnerships with all people of this province as we work towards true reconciliation and honor this beautiful land together. So today is our last seminar of 2021, and I have the pleasure of introducing our final speaker, who is Dr. Gerry Wright. Gerry has a PhD in chemistry from the University of Waterloo and has postdoced at Harvard Medical School, where he worked on molecular mechanisms of antibiotic resistance. He has been a professor at McMaster University since 1993 and holds the Michael G. DeGroote Chair in Infection and Anti-Infective Research and a Tier 1 Canada Research Chair in Antibiotic Biochemistry. And today he will be talking to us about his work on the ecology and evolution of antibiotic resistance and new tools to explore diversity in microbial communities and applications to medicine. So welcome, Gerry, and you can go ahead and share and take it away.
Okay. So thanks, Levi. Thanks for the invitation to, let's see, am I sharing the right screen? Doesn't look like it. Hold on a second. There we go. That's better. Yeah, so thanks for this invitation. I've been meaning to get out to Memorial for many years now. Kapil, Talon, and I have been conspiring to get me out there, and I guess it's not quite the same thing as being there in person, but I'm delighted to be here, at least virtually, and happy to tell you about some of the ways that we're thinking about how antibiotics and antibiotic resistance sort of integrate with its natural history, and hopefully you'll find it entertaining. We'll see. I just want to start out by explaining why we in my lab work on antibiotics, and so I'd like to really make the case that antibiotics that we all take for granted now in 2021 really have changed the way that we die. So if you look at the pre-antibiotic era, this is 1920 Statistics Canada data, about all of the causes of death, about 56% of those were associated with infections. The life expectancy of a Canadian 100 years ago was just under 60 years. So if you look at the latest data that we have available from StatsCan 2018, you know, now the causes of death are not primarily infectious disease. This is pre-COVID data, where there's going to be a little blip, certainly over the last couple of years. But really, we're down to really small numbers, 3% of death caused by infectious diseases. Most of the way that we die is from just diseases of old age, where we're just getting worn out. So cardiovascular disease, stroke, Alzheimer's, now chronic diseases such as diabetes. And we've gained over 20 years of life expectancy. So when we don't have antibiotics, we lose all of these amazing gains. And so this is the reason why we study antibiotics and antibiotic resistance in my lab. So this is my history of antibiotics. We discover them, we're very, very happy. Time goes by, and then the microbes become resistant to it. Again, this is important to realize, it's the bacteria that are becoming resistant, not us. But that's okay, because we're very good at finding new antibiotics. So we just keep repeating this cycle, and that has worked incredibly well since we first brought some of the first antibiotics to market in the 1930s, the sulfa drugs, and that was followed by penicillin in the 1940s. And this cycle worked very, very well for about 50 years, but it's now completely broken. We're not discovering antibiotics in a fashion that's fast enough keep up with resistance, and resistance is only growing. So the question you could ask yourself is, why is this happening? Well, there's two major reasons. One is economics. The economics of drug discovery, in particular antibiotics, are just not favorable to getting into this field. So if you're we rely on large pharmaceutical companies and biotech companies to deliver new drugs to us. We've seen that just in lightning speed during the time of COVID. But in the case of antibiotics, because of the way that we pay for them, it's just simply not cost effective for large pharmaceutical companies or biotechs to be working in this area. So they've really retreated from it. That's not something I can solve, or likely more that you can solve. This is something that government policy has to solve. And we're working pretty hard to do that. So I'm hopeful that could be solved. But the science is challenging, and the science is, if it was really easy to find antibiotics, we'd have lots of them. And so what are the scientific barriers? Well, resistance is a massive problem, and that's what I'm going to focus on today. But there's a lot of other reasons, and one of them is the failure of target-based drug discovery approaches that really dominate the current strategies of pharmaceutical companies about how we find drugs, in particular for human targets. It just doesn't work for bacteria. And one of the major reasons for this is because the libraries that work so incredibly well, the chemical libraries as starting points, that work so incredibly well to find new blood pressure medicines or antidepressant drugs, simply do not work as starting points for antibiotics. So these things really change the landscape, but it's resistance that I'm here to talk about today. And my reason for doing so is as outlined here. And I think we cannot get in front of this problem, even if we can solve the economic issues, and even if we get better chemical libraries and things. If we do not understand where antibiotic resistance comes from, what its genetic and biochemical diversity is, and how the evolutionary forces are applied to it in order to enable resistance to emerge so predictably in pathogenic bacteria. And I think one of the reasons that we struggle with this is because microbiologists, and this is no offense to the microbiologists listening, is that microbiologists are just not good at a few things. One of them is thinking at the dimensions that bacteria live on. So microbes, we tend to think of them, we plate them out on a petri dish, or we do a minimal inhibitory concentration screen in liquid media, and we think about them in pure culture, but they don't live that way. They live in complex consortia, whether it's in your gut or in your bloodstream or in the soil. And they interact with each other at the micron level. So the dimensions that we're thinking about are really small, and diffusion becomes a really significant challenge. And as you're thinking about how resistance and drug discovery, you start having to you really need to start reducing your ideas to the way that microbes interact with each other. So that's one thing. That's the very small. The other thing that we're not good at, and I count myself in this, is thinking on the time scales that microbes work on. And these are geologic time scales. So this is the history of the world, formation of the earth about 4.5 billion years ago. We see the origins of life around 4 billion years ago, and then the first bacteria emerge a little over 2.5 billion years ago. And the first or the most recent common ancestor of E. coli and Bacillus is about 2 billion years. It's an enormous number that we don't, it's hard for us to fathom. If you're a geologist, you're thinking like this all the time. If you're a microbiologist, you tend to think that, you put your E. coli in and it'll be all grown up the next day. The eukaryotes come on the scene a few 100 million years later. And if you start looking now at the closer era to us, before even Pangea breaks apart, we see dinosaurs and us over here, a little pixel on this side. So these are the kinds of time scales that we need to be thinking of, the evolution of microbes and their genes and their development of the ability to evade antibiotics. over this time. So it's hard to think on those scales. And the other thing is just the vast microbial genetic diversity on the planet. So the estimate is that there's 5 × 10⁻30 prokaryotic cells, so 5 × 10⁻30 prokaryotic genomes on the planet. You know, there's 10⁻17 seconds since the Big Bang, right? There's only 10⁻21 stars in the entire universe. This number dwarfs those. This scale of microbial genes on the planet is stunning. So you have to think that the microbes are working on scales that are really small. They've been around for an incredibly long time, and there's just this vast genetic diversity, genomic diversity to access. And this just becomes, so then in this context, resistance becomes inevitable. And so the other thing that we need to start thinking about is just the amazing chemical diversity. So these microbes have been around since on the planet for billions of years. The way that they interact with each other at those scales is through the development of very diverse chemicals. Some of these are antibiotics, some of them are siderophores to acquire iron and other metals. Some of them are communication tools where they can talk to their neighbors and themselves through things like quorum sensing. But there's encoded in those 10⁻30 genomes on the planet is just an absolutely unfathomable chemical diversity of molecules that are tailor-made to interact with bacterial receptors. So all of this means that where these molecules are toxic, resistance, when you start thinking about these scales, thinking about time, it simply becomes resistant or inevitable, sorry. And many years ago, we started thinking about this and did a little study where it's almost a high school science fair study, frankly, where we went out to the local areas around the McMaster campus, and we went to some agricultural sites, and we went to some sites in Northern Ontario where there's not a lot of people. And we took some soil samples and we isolated about 500 soil bacteria and just asked, are they drug resistant? And to our surprise, although when you think about it in retrospect, it probably shouldn't have been surprised, but they were resistant to, on average, 7 or 8 different antibiotic classes. And this includes natural product antibiotics that are made by microbes. It includes semi-synthetic derivatives of them, and it includes completely human-made synthetic chemicals. It didn't matter. All these organisms we could detect resistance to them. And a few years later, Gautam Dantas, who was then in George Church's lab, found that indeed it didn't take too much effort to find organisms that grow on antibiotics as their sole carbon and nitrogen sources. So this is a reflection of the fact that microbes have been on a planet for such a long time, and they're used to interacting with these kinds of toxic molecules. So when we did this study, we had a little bit of pushback and said, well, we live in the Anthropocene. There's probably a very good chance that a lot of microbes that you were studying had some kind of a previous exposure to human antibiotics. Are you really sure that this is not due to some prior exposure to antibiotics? And I didn't think it was a fair comment at the time, but it was a comment that some people made. And so we were thinking about it and saying, well, where on the planet could we find environments that are completely pristine, that we can be sure have not been affected by humans? And it's hard to do. Whether it's the top of Mount Everest or the bottom of the Marianas Trench, there's evidence of human activity that's really easy to find. And then I happened to go to a meeting, so the importance of going to meetings. I happened to go to a meeting where I saw a talk in an area that I knew nothing about, which was cave microbiology. And that introduced me to Hazel Barton, who's here, who's an amazing scientist and also a caver. And she had access to this cave called the Lechuguilla Cave. It's in New Mexico. And the thing that's special about this cave is that it's the deepest and largest, most extensive cave system in all of North America. But it has been isolated from the surface for 3 million years. So it wasn't discovered until the late 1980s when cavers sort of thought that there might be something on the other side of a rock wall, and they actually bored a hole into it and put a big sort of submarine style door on it. So there's no access of this cave to the outside world for millions of years. And there's no bats in this cave. There's no blind fish in this cave. There's no, typical things you see in other kind of caves that are open to the surface. Instead, the only living organisms in this cave are bacteria that have crept in over millions of years, and they live on biofilms along the sides of these large surfaces. And so, with Hazel's help, we were able to collect about 100 of these, and we did the same kind of study, this time looking at gram-positive and gram-negative bacteria against 20 or so different antibiotics. Again, the same kind of spectrum. And again, we saw the same thing. Lots and lots of resistant organisms, no antibiotics were spared. And so this really told us that antibiotic resistance is hardwired into the bacterial genome. So these bacteria, though, of course, are not old bacteria. They're contemporary bacteria, just like the bacteria that we isolated in soils. And so you started to ask ourselves, well, if this is what the state of the universe is now, these bacteria are alive today, but they've been isolated from the surface for 3 million years. This does suggest to us that resistance is quite old. And that was the subject of a few other studies that I want to share with you. So the fact that it's everywhere would suggest that it's been around for a long time, well predating human use. So in one study, I want to take you back to Troy. We've all heard the story of the Trojan horse, when the Greeks built it and during their siege of the city in the Hellenic times. Troy is a city in what is now Turkey, but has been continually inhabited for hundreds of years, thousands of years. And so I'm going to take you to Byzantine Troy, so late 13th century, about 900 years ago. And some archaeologists that not our lab directly, but some colleagues were interacting with, discovered a grave of a young woman who died. And when they were uncovering this skeleton, what they found alongside of it were these nodules, and they were interested in the fact that maybe this is an evidence of tuberculosis. And so they contacted Caitlin Pepperell, who's a tuberculosis expert at the University of Wisconsin at Madison, and she contacted Hendrik Poinar, whose office is right beside mine, an ancient DNA expert who's been working on Neanderthals and mastodons and mammoths and what have you. And together with him, we were able to isolate DNA, 900-year-old DNA from this nodule. And what we found was human DNA, as you would expect. We found that the DNA associated with this young woman. We also found a Y chromosome. So that indicated to us that there was this woman was pregnant at the time with a male child, infant, fetus. We found not tuberculosis DNA, but Gardnerella vaginalis, which suggested to us that that this was actually a nodule associated with inflammation or an infection in the uterus. And the most of the genome that we are isolated was from this opportunistic pathogen called Staphylococcus saprophyticus. And we were able to get most of that genome assembled as a result of this. We're able to explore its evolutionary history. But the thing that's relevant to this story is that it has in this genome, it has a beta-lactamase. Beta-lactamase is, as I'm sure you all know, and hydrolyzed beta-lactam antibiotics. This is ampicillin, cleaves this bond. And we were able to resurrect this enzyme and show and study its enzymatic activities. And for the biochemists in the audience, you will recognize this rate constant. This is the second order rate constant. And its number here of 1 times 10 to the minus times 10 to the 8 suggests that it's limited essentially by diffusion. So this is the hallmark of beta lactamases that are active in the clinic today, which basically means they are supercharged. This is what they do. They are highly evolved catalysts for drug inactivation. And here we find it 900 years ago in this patient. So these resistance genes have been around for a while. And we were able to reach back even further by exploring the metagenome in 30,000-year-old permafrost. Again, this is with my colleague Hendrik Koynar, with some collaborators at the University of Alberta and the Yukon territories, and who this team studies the permafrost, who I'm sure you're aware is the permanently frozen sediment at the top of northern regions, in particular Canada and Russia and other places in the Nordic countries. The Canadian permafrost in particular is especially well preserved, and we were able to isolate and very carefully date core samples from 30,000 years ago, brought them back to the lab at McMaster. We have an ancient DNA lab, and then when you work in an ancient DNA lab, it's almost like working in a containment level 3 lab. Everyone's all gowned up to make sure that we are there. Our concern is not infection, but rather contamination from modern samples. And so using very, very careful ancient DNA techniques, we were able to isolate the metagenome, and we could easily find samples of megafauna at the time. So woolly mammoths, longhorn bison, miniature horses. And we also found genes for resistance to penicillin, to tetracycline, and to this very complex antibiotic, vancomycin. So this is physical evidence of the ancient origins of antibiotic resistance that continue to plague us today. So using ancient DNA techniques, we can probably go back another 50,000 years or so, but after that it gets a little challenging, and just technically, just because the DNA is so fragile. But we did wonder, we're sitting around after we did these studies, is can we look back? Is there a way for us to look back even further? And there is, not with direct samples, but through inference. And so this is Nick Waglechner. He was a PhD student in my lab, an amazing bioinformatician, and he was interested in how this very complicated antibiotic called vancomycin that I just showed you was actually synthesized. And so antibiotics like vancomycin are natural products produced by microbes. When they do so, they assemble all of the genes that are necessary to make something like vancomycin collinearly on the chromosome of the producing organism. We call these natural product biosynthetic clusters. And this very complex compound, you could actually break it down to think about what do you need to make it. And so Nick did this. And if you're a chemist, you would do something called a retrosynthetic analysis. And so you would say, well, what you can think about is start stripping away the decorations on here, the sugars and the hydroxyl groups and the chlorines. That would give you a cross-linked peptide. This is really the thing I forgot to mention. These are peptides that are highly cross-linked to give them an incredibly rigid three-dimensional structure. It allows them to bind to the dealanine terminus of the cell wall. It's an amazingly well put together host guest system. So continuing our retrosynthetic analysis, that cross-linked peptide is derived from a linear peptide, and those linear peptides are made up of amino acids. In the case of vancomycin, there's some proteinaceous amino acids, like in this case leucine, but there's also a lot of weirdos like phenylglycine and dihydroxyphenylglycine and hydroxylated tyrosines. So using this, Nick started to look in our rather extensive database now of antibiotic producers, and it's a whole other part of the lab that I don't have time to talk to you about today. It's looking for the clusters that might make glycopeptide antibiotics, and also to the available genome sequences that are freely available on public databases. And so he collected about 80 of these biosynthetic gene clusters, and he started building family trees for all the individual genes that are common to the synthesis of vancomycin-like antibiotics. There's lots of them out there. And so he would make a family tree for the peptide synthetases and the amino acid synthetases and the sugar transferases. And he built about 200 of these phylogenies based on the genes that are associated with glycopeptide antibiotic gene clusters. And then he reconciled them with the family tree of the organisms that actually produce them. So these are actinomycete bacteria. This phylogenetic analysis has been dated by Cameron Curie's group, by a PhD student at the time, Mark Charette. So Mark and Cameron were able to actually date the emergence of this really important class of antibiotic producers. And so with our phylogenetic analysis reconciled with this, with the timeline associated with the emergence of the producing organisms, we can actually reconstruct how and when glycopeptides antibiotics actually came to be put together. So let me walk you through that. So about 1 billion years ago, the genes associated with a lot of the biosynthesis of the individual amino acids emerge, as well as some of the sugar synthases. Somewhere between 1 billion and 500 million years ago, all of the enzymes that are required for making the peptide, these are found on assembly line mechanisms called non-ribosomal peptide synthetases, and the cross-linking enzymes come together. So we think that by about 500 million years ago, something that resembled a glycopeptide was out there. What was missing in this, of course, and what I failed to remind you of, is when you have a biosynthetic gene cluster to make an antibiotic, you also have to have resistance genes, right? So a lot of the resistance that we studied in these soil microorganisms that I spoke to you at the beginning of the talk really come from producers. And so these resistance genes are essential, because otherwise you would make your antibiotic once and you would immediately poison yourself and then you would die. So you have to co-evolve resistance genes. So these resistance genes start to emerge about 350 million years ago. And so then we start seeing radiation across what we now believe to, or what we now know to be the glycopeptide diversity that we see today, where there's literally hundreds, I'm sure thousands of different variants of these kinds of molecules out there. So it took about 500 million years to get everything in ready to go. And then once the resistance gets linked up to the biosynthesis, you can have this explosion of chemical diversity over time. So glycopeptide antibiotics are at least 350 million years old. And it's sobering to realize that it probably took less than a decade for us to see emergence of resistance in the clinic. So what took nature, 500 million years to assemble, and then 350 million years of success on the planet, we kind of ruined for ourselves in a decade. If we go back to that graph that I showed you, that you can, if you're interested on, you can find in this review that we wrote, where here's the dinosaurs. We can now start to add resistance genes. So there's been some similar studies on beta lactamases that are probably at least 2 billion years old. Some metallo-beta lactamases are about 1 billion years old. And here's our glycopeptides. Glycopeptides have been around well before the dinosaurs. Gives you some kind of idea of where we're at. And a similar study was done with streptomycin. So these are really ancient molecules that have been around, again, for a really long time. So that speaks to the time part of it. And I think it's really sobering to think about how successful these have been over the years. I want to switch gears a little bit now and just take you back to the cave, because the organisms that came out of the cave were quite fascinating to us in the lab. And Andrew Pulowski, who was a PhD student at the time, is now a postdoc in George Church's lab, decided he was really interested in this one organism, called Paenobacillus in Lechuguina Cave 231, a very clever name. And what he found is that it was resistant to 26 of 40 antibiotics that he tested. And in particular, one that we found really interesting, which was daptomycin, which is the sort of latest antibiotic that has been a completely new scaffold that was brought to market in the early 2000s. But it was resistant to all these other antibiotics, some of which we know, and some of which we had not seen resistance to, like bacitracin. So what Andrew did is sequence the genome of this bacillus, and then he plopped it into a platform, a bioinformatic platform that we have called CARD, the Comprehensive Antibiotic Resistance Database, that I would encourage you to look at if you're at all interested in this kind of stuff. There's a tool on CARD that enables us to input the genome and then ask what are the antibiotic resistance genes that CARD recognizes and find them. And so what he found is that out of those, out of the 26 antibiotics resistance phenotypes, using CARD, we could predict which genes were associated with it. And in fact, Andrew went and cloned all of them and showed that indeed they are bona fide antibiotic resistance genes. But then there was a bunch of resistance phenotypes not associated with known resistance genes. And to study those, Andrew used functional genomics, essentially just shotgun cloning of the entire genome and then selection for resistance. And I'm going to show you one story very quickly. And this is bacitracin, an antibiotic that was discovered in the 1930s, actually, by René Dubos from other bacilli. And this is bacitracin. Is again another remarkable organism. It's antibiotic. It works by binding to undecaprenyl pyrophosphate. So you might remember that that's a lipid phosphate carrier that brings the precursors of peptidoglycan synthesis from the inside of the cell to the outside. It's like a lever. And what bacitracin does is it doesn't get in the cell, but when this pyrophosphate is sticking out, it wraps itself around it with the cooperation of some metal ions. But a key interaction is through this asparagine residue right here that forms an interaction with the terminal negatively charged phosphate. And what Andrew found is that you could get resistance to this, and resistance had not been reported before in this organism. So this is the sensitive sensitive bacitracin, and they incubate it with the organism, and then you end up with this resistance phenotype. And if you purify the compound, it's only changed, its molecular weight is only changed by 1. So it's not like it blew it apart or anything. And what he found is that what was happening is that there's an enzyme called BAHA in the membrane that converts this asparagine residue to an aspartic acid. That explains the molecular weight change. And it also explains resistance, because an aspartic acid now is negatively charged. So negative charge, they no longer want to interact with each other, and you get resistance. So you have this remarkable new mechanism that we found in this organism in the cave. And then Andrew went and found that indeed there are surface organisms that have the very, very similar mechanisms. And so it really is a remarkable response to this, as I said, this amazing chemical diversity that exists out there. And then it's counteracted by an equally diverse and exciting, interesting biochemical and genetic diversity of resistance. And that led us to think about this in a more holistic way. We developed this concept called the antibiotic resistome. So if you do a PubMed search of resistance, what you really get back is just a lot of discussion of resistance in the clinic. And that makes sense, because that's where we tend to care about resistance, because we use antibiotics as drugs. But clearly, that's not the story of resistance. In fact, resistance in the environment that is outside the aegis of pathogenic bacteria is much more diverse. We have the contribution of antibiotic producing bacteria, as I mentioned to you before in the case of vancomycin. But we also have all of this other, what we're calling sort of cheekily cryptic resistance. So all of the, the painted bacillus is resistant to bacitracin, but it doesn't produce bacitracin. And then what happens then is through the use of antibiotics in the clinic, you have this selective pressure for gene capture by mobile genetic elements like transposons and phage and even just natural transformation from this gene flow going from the environment to the clinic. So that helps us explain why we have so much resistance in the clinic right now. But I want to just finish the last few ideas in the talk to talk to you about where do these highly evolved resistance genes come from in the first place? And we call this, or we think about this in terms of proto-resistance. So in the analogy is proto-oncogenes for us. And the way we think about it is the following, is that in order to get, in order to evolve a resistance element, you have to have some precursor protein, and it may have no resistance at all, or it may have a little bit of resistance. It's not very efficient. But then with selection, and again, remember, we've got time on our side in terms of thinking about geologic time. You have antibiotic selection, and then eventually you end up with this very highly evolved, very efficient machine whose job it is to inactivate antibiotics. So how do you study this? We use an approach that was described by John Gerlt at the University of Illinois, called genomic enzymology, in which we, characterize the enzymes using biochemical means, determine their three-dimensional structures, identify homologs inside of genomes that are available, and sort of iterate around here. And let me give you a couple of examples. And talk a little bit about resistance to the rifamycin antibiotics. Rifamycins are natural products derived from soil organisms, but they're by themselves, the natural products are not great antibiotics. But when you modify this naphthalene group with some semi-synthetic approaches, that is doing some chemistry, you get really amazing drugs like rifampin, which is a WHO essential medicine used for the treatment of tuberculosis, for example. So the way that the rifamycins work is they bind to RNA polymerase, and they don't bind in the active site to jam up its activity. They bind to the exit tunnel where the newly formed RNA is coming out. And again, you have, there's a theme here, right? You have these very complex compounds. This is what it looks like in 3 dimensions, where it's actually got like a little naphthalene seat, and then there's this bridge, which is this polyketide over here. All of this complexity is designed just to position 4 hydroxyl groups in exactly the right spot so that they can interact with very common residues in the exit tunnel of RNA polymerase. And so all of this complexity is just to properly position 4 hydrogen bond donor receptors in 3-dimensional space to exactly cassette into this site, and that's how they work. You can get resistance through a number of different ways, but we're really interested in enzymes in my lab, and you can get resistance through it. This is like a champion. There's ADP ribosyl transferases, there's sugar transferases, there's kinases, and then there's even monooxygenases that result in decomposition. And we've studied all of this, but I just want to talk about phosphorylation for a second. And one of the reasons for it is because we've been studying antibiotic phosphotransferases for many years, and we were the first to show that antibiotic phosphotransferases that confer resistance to aminoglycosides and macrolide antibiotics, so aminoglycosides like gentamicin and macrolides like erythromycin. were derived from protein kinases, serine, threonine, tyrosine kinases. They all have the same three-dimensional structures, as you can see here. They have the same overall mechanisms. They share the same active site geometry, everything. So they all have a common ancestor, there's no question. So I had a graduate student start up in the lab, Peter Giannopoulos, and I said, well, we know that there are these rifamycin kinases in some of our samples that we looked at. Why don't you go and find it? There'll be a good first project that you in the lab. It's going to look like these known kinases, you know, you'll have a good first paper to start your graduate career. And this didn't work at all. Peter tried incredibly hard using a number of different technologies to try and find these kinds of kinases. In fact, he sequenced genomes, and at one point he actually expressed 45 different candidates that had this sort of same kind of structure, and none of them inactivated rifamycin. Instead, what he stumbled on, and this is entirely due to him, this is Peter, was that the culprit were part of a family that are similar to dikinases involved in the biosynthesis of phosphoenolpyruvate, so primary metabolism enzymes. And and I don't have time to tell you the whole story about how we got into this. Is really an amazing, and entirely a beautiful example of the graduate student not listening to the supervisor, because I told him to give this project up, but instead he opened up a brand new area of biology that nobody knew about before. So what he found is that these rifamycin kinases were related to these dikinases. And what he found is that, you know, the mechanism is phosphorylation at this hydroxyl group. And you remember this hydroxyl group is essential for interacting with the RNA polymerase. And so by phosphorylating it, you get resistance. And then again, with Nick, he started looking at similar kinds of proteins and found them in a number of different genomes, or sort of similar genes, and found them in a number of different genomes, including in Listeria. And we decided to study the one from Listeria because it actually, frankly, it crystallized very well. And so Georgina Cox, who was now assistant professor at the University of Guelph, but then a graduate student in my lab, a postdoc in my lab, and Peter Stogios, a student in Alexei Savchenko's lab at the University of Toronto, solved the crystal structure. It has this amazing sort of three-lobe structure, an ATP binding domain, a rifamycin binding domain, and then this swivel domain. And the key to this mechanism is that the ATP binds, it transfers not the gamma phosphate that is normally found in kinases, but an entire pyrophosphate. So you have this histidine attacks this beta phosphate. You form this pyrophosphate, transient pyrophosphate intermediate. You lose one of the phosphates that happens to be the gamma phosphate. And then you have this histidyl phosphate. And then the whole thing moves from one position to the other, and you get transfer of 40 angstrom movement of this protein, of this histidyl phosphate onto the rifamycin antibiotic. And that's how you get resistance. So it's not a simple just transfer of, like we saw before in the other antibiotic kinases. This is an entirely different machine, really, really interesting. And so if you're thinking again about where these things come from, you have this ancestral protein that likely is the direct precursor to these phosphoenolpyruvate metabolizing enzymes that was co-opted to result in rifamycin resistance. So this is an example of how, you know, you can use primary metabolism as a starting place to move your catalyst or your enzyme towards resistance. And I just want to finish up with a story we just published on a variation of this, where you take a resistance gene and then you give it a new function. And this is associated with the development of this antibiotic. This is called apomycin. Apomycin is an aminoglycoside antibiotic. Aminoglycoside antibiotics have been around for a really long time. There's lots and lots of resistance to them, mostly through enzymes. that either I mentioned kinases, but there's acetyltransferases, there's nucleotidal transferases, and lots of different ways you can become resistant to antibiotics like tobramycin. There's literally thousands of these resistance genes in CARD, if you look at it, or hundreds in CARD and thousands out there. Drug discoverers at an antibiotic discovery company in the early mid-2000s called Achaogen said, well, we can take this information, we can build a better aminoglycoside so that it only has a couple of liabilities, this acetyltransferase and this kinase, but it's not affected by all these other antibiotic resistance enzymes. And they were very happy with themselves. But while they were developing this antibiotic, a new resistance mechanism emerged in the clinic, and this is a methyltransferase that modifies the ribosome where aminoglycosides bind. And so this antibiotic they worked very hard on was actually still susceptible to this new mechanism of resistance. Apomycin is interesting because it is not. In the literature, it really only had one liability, this one acetyltransferase. But then while this was being developed, this paper was published. Oh, I missed that. Oh no, where is it? Where is that? There it is. Where we saw the identification of a new resistance gene called APMA in Staph aureus that was associated with food animals. And this gene is interesting because it did not look like any other aminoglycoside resistance element. Instead, what it looked like was a member of a family of enzymes that are associated with the inactivation of chloramphenicol and virginiamycin antibiotics. These are O-acetyltransferases. They belong to a family called, excuse me, the left-hand beta helix, that's what this is here. So this L-beta-H superfamily. So you form these trimers of these monomers, like this is what they look like. You can see the left-handed beta helix over here. And the active site is at the interface of these trimers. It's really quite cool. And these are O-acetyltransferases. All of the aminoglycoside inactivating acetyltransferases are N-acetyltransferases, and they belong to a totally different family called the GNAT superfamily that are associated with things like histone acetyltransferases, all N-acetyltransferases. So we were interested in this. And then, so Emily Bordelot, a PhD student in my lab, started working on this family or on this enzyme, APMA, and she crystallized it and found indeed that it is a left-handed beta helix protein, just like you would expect. It has this really important catalytic histidine that is essential for the formation, for the activity of things like chloramphenicol acetyltransferase. So in these O-acetyltransferases, what this histidine does is it acts as an active site base to pull off this proton, because it's hard to, the pKa of these protons is something like 10 or 11. Hard to just go ahead and modify this with acetyl-CoA. So there's a pretty complex mechanism where the enzyme active site is positioned to make this hydroxy group very nucleophilic. But what Emily found is indeed what was happening with apomycin is it was being transferred to a nitrogen, not an oxygen. But that this histidine was still really important. And so what she found, again, using a whole series of crystal structures, was that the histidine is there now not to act as an active site base, but to actually position, steer the antibiotic in exactly the right geometry so that you can modify this nitrogen with an acetyl group. So here's an example. The previous example of a kinase was modification of primary metabolism to give you resistance. This is an example of modification of an existing resistance element to give you something that does things in a completely different way, but still requires the same kinds of active site tools. So the take-home message in all of this is that there's so much to learn about resistance by studying these biochemical mechanisms. There's so much diversity out there. And the reason for it is because these microorganisms have been living with each other for billions of years. They have been interacting with each other through these very complex chemistries for billions of years. They have developed means to amplify and to dampen these signals through things like resistance. They've captured biochemical mechanisms from things like primary metabolism, or in the case of the vaporomycin, as I showed you, from other resistance genes, and through selection have generated bona fide resistance genes. And the thing that's happening in our lifetime is that these genes are moving from this vast, diverse ecology that exists around the world into organisms that cause diseases in humans and animals. And that's resulting in the reduction of our antibiotic efficacy. And to go right back to our first slide, jeopardizing the extra 20 plus years that we've gained as a result of our ability to control infection over the last century. So I think it's really important to study this. Where do these things come from, so that we can be better prepared for the antibiotics of the to deploy the antibiotics of the future. So I'm going to stop there. I want to thank all the people that funded the work in the lab. These are the current ones. This is the team about a year and a half ago, all masked up, as I'm sure all of you are. And again, I want to thank you for the invitation to speak to you, and very happy to take any questions if you have them.
Excellent. Thank you very much, Gerry, for the talk. I think, so what we'll do here is if anyone has a question, please either indicate so in the chat, or feel free to type out your entire question in the chat and we can read them out for you. Okay, Andrew, go ahead.
Has a question.
Hi, yeah, thanks very much. It was a really great talk. I thought the part where you traced the evolution of the one synthesis of, I think it was vancomycin, was really cool. So is there any evidence you can find, like maybe in, I guess you can't look that far back in the record to see, like, did that coincide with like a proliferation or like an enhanced success of those organisms, or you think like they maybe colonized a new niche or something like that?
Yeah, that's a really good question. I don't know. Clearly, it offered some kind of strategic advantage, because otherwise we would not have the diversity we have today. And there literally is, the more we look at genomes, there's thousands of these. So they must have played a role in some kind of success story, but it's hard to, as you said, because now we're not looking at the fossil record or anything. We're just looking at sequence gazing, and so it'd be a matter of trying to start to triangulate some of this diversity. Although, if I do go back and look at, I'm not going to bother showing you this; it's too boring. But a tree. Sorry.
I guess the fact that it proliferated as a trait means it was obviously helpful, right? I guess that's maybe the...
Yeah, so if I'm looking there, that it does coincide very closely with the sort of root of the actinomycete family. And so, yeah, you're right. So actually the answer to your question is yes, it must have been pretty important.
Great, thanks very much.
So, yeah.
Thanks again for the talk.
My pleasure.
Okay, Dawn, can go ahead.
Hey, Gerry.
Hey, Dawn.
Thank you so much. That was a fantastic talk. In terms of just thinking about like all the different mechanisms of resistance, so not only modification by enzymes, but even like efflux mechanisms, is there any evidence as to which particular mechanism evolved first? I don't know if that's something that...
Yeah, that's a really good question. I'm going to guess that efflux is probably the champion.
Right.
If I was to pick my favorite if I had to just go out to Mars and not have, and just pick a couple of resistance mechanisms to have if I was a microorganism, I would pick efflux.
Yeah, I would have
The
guessed the
enzymes
same thing, but I mean,
Yeah.
I don't know if it's something that somebody's looked at in detail to try and actually figure it out.
No, it's a really good question, and I think there's a lot of really interesting things about efflux, and we don't study efflux. We should probably. I'm really interested in enzymes, and so that's why I focus on. But you know, the efflux systems are beautiful examples of non-specific resistance. There are some very specific, enzymes tend to be specific for a class, even to a specific member of that class. The efflux systems are amazing because it's kind of like the innate immunity of bacteria, right? They're very generalists. Right. They tend to be, especially in the case of gram negatives with the R&D tripartite systems where they, you take an organism like Pseudomonas or Burkholderia that lives in the environment. They're armed to the teeth. They're just absolutely not having any nonsense from anybody in terms of toxic molecules, and it makes it so challenging. So I would definitely guess that efflux came first and make the most sense to me.
Right, absolutely. All right, thank you.
It's a great idea. We should actually try and date it.
Yeah, no, it's just as you were going through your talk, that was sort of what popped in my mind. So, all right, thank you.
Thanks, Don. Nice to see you.
All right, Kapil has some.
Kapil.
Hi, Gerry. How are you?
I'm doing great. How are you?
Great, excellent talk. And as usual, I always leave your talks with more questions and makes us think more about stuff. So the whole idea about glycopeptides and beta lactams, and it's we've pondered about it for a while. Which one do you think is older? Like because the beta lactams?
Yeah, for sure. It's a really, really ancient biosynthetic machinery. It only takes 3 enzymes to make a beta lactam, the core beta lactam ring. It's really, and there's been a fair bit of work on the beta lactam, the ACV synthase, which is the core beta lactam production system, is at least a couple of billion years old. Yeah. And it's and so there was some work done. Actually, some of the work that totally inspired us to do our study on glycopeptides was done in the early 2000s, looking at this, using molecular clocks and things like that, and that's where we were able to position the beta lactamases on that history. So they're definitely the winners.
Yeah. And the other cool thing was in Andrew's work where you found those resistance genes from Paini bacillus. There was linozolid resistance in there.
Yeah.
And
yep.
Did he have any?
So that's So that's a methyltransferase that modifies the ribosome. Okay.
And not related to other mechanisms and pathogens, or?
Yeah. So there's and that's one of the things that there's a postdoc in my lab, Adam, who's working on that family. A really interesting. There are radical SAM enzymes that modify the bases, not on. There's some ribosomal methyltransferases out there, including the one that I made reference to for aminoglycosides. They modify exocyclic nitrogens on adenine, but this methyltransferase modifies that carbon. So it's one of these really weird radical SAM enzymes, and they're emerging in the clinic associated with linazolid resistance in gram-positive pathogens. Yeah.
Okay, and I guess just one more. I know there's a student waiting in line. So this kind of also brings us, like, we had a similar story about repurposing resistance for biosynthesis. We're working on with the beta lactamase. We've where we have this beta lactamase with isomerase and a cyclase domain on it. So do you think we need to start thinking about the resistance genes in gene clusters as just being resistance, and they actually might just be repurposing the pathway for making something we haven't seen before? because, you know, many times we just go that, oh yeah, that's a self-resistance gene, just drop it.
Yeah, I totally agree. Yeah. And actually we've got an example of that that we're just about to report. Actually, it's not quite the same, but in this case, there's a self-resistance gene that modifies this really broadly impact, a really broad anti-cancer compound that we've been studying, that when you do that self-resistance mechanism, it actually completely tailors it down to cancer stem cells. And so you get this gain of function through bacterial resistance that is completely unexpected. So I think all of these, I don't think take anything for granted anymore. I think there's a lot of, there's so many cool stories out there.
All right, thanks, Gerry. Thanks again. I think there's a student waiting right there. Yeah.
Right, Arshad.
Thank you, Dr. Wright, for the talk. It was really fascinating.
Thank you.
So, like, by looking at the talk, I just got to know, like, I knew, but it was some kind of evidence was showing that we are really way behind in terms of antibiotic and antibiotic resistance. So my first question is like, how understanding resistance we can go for discovery of antibiotics, or how can we go for a better approach to stop antibiotic resistance, or to prevent it somehow, or to take precautions against it?
So I'll give you a simple answer, then I'll give you an example. The simple answer is just, you know, there's that old adage, right? Know your enemy. So we need to really understand resistance, in particular, as we're moving natural product, and there's a lot of efforts to do that, natural product antibiotics into the clinic going forward. You know, it's pretty doom and gloom right now in terms of development, but there's still lots of people working on at the discovery element. So I think we just need to, you know, had we done the kinds of studies that I'm describing, say with vancomycin, and there was stuff in the literature that said the way that it worked, there's probably no resistance, right? Which is completely silly. We would have known what to look for, you know, in terms of surveillance and things like that. So I think there's knowledge is power in this case. So you have, the more you can learn. But let me give you a very concrete example where we used resistance, actually the lack of resistance. So the glycopeptide story that I told you, so that started as this very gee whiz. We're sitting around, you know, how old are these antibiotics? Can we actually use this phylogenetic approach to date it? And it was entirely, we have a lab retreat once a year, well, we did until COVID, where we all go up in Northern Ontario to, you know, some cabins, and we sit around, we talk science for 3 days. And thinking about where the future of the lab is going to go. And we started talking about these kinds of big questions, like, where, you know, can we use phylogeny? Can we do genome mining? Can we figure this out? And that's how this project started. And then when Nick built those phylogenetic trees, what we were able to find is that they clustered into branches associated with resistance. And then there were parts of the tree that were not associated with typical vancomycin resistance. Right, we had pulled these genomes out of, because they looked kind of like peptides. They were all obviously related, but they weren't necessarily just 10,000 flavors of, or I'm exaggerating, but, you know, 80 flavors of vancomycin. And so we found as 2 branches on the tree that were not associated with typical resistance. And we found a new class of antibiotics that works in a completely novel way. And we just reported that a year ago. There's in a Nature paper where they look similar to, obviously, because they have a lot of the same biosynthetic machinery, but they don't have the same resistance mechanisms because they don't work in the same way. Instead, what they do is they bind to the peptidoglycan and they inhibit the cell division by inhibiting access to the peptidoglycan by autolytic enzymes that are essential for pinching the peptidoglycan apart. So brand new mode of action, no significant resistance that we can find. There's got to be some kind of resistance mechanisms out there, but we haven't found any yet. So by using the absence of resistance to help drill down, we were able to find a new class of antibiotics. So yeah, it's all about how you sort of creatively apply what you learn, right?
Right. And another question is like in the bacteria we found Penibacillus 231. We found like 26 out of like antibiotics
Yeah.
was resistant, and you have already like kind of answered the question, but like, was there any other gene or any other resistance mechanism in that bacteria which was like completely different and which we don't know about? You know, because it is showing 26 it is showing resistance against 26 antibiotics. There could be a possibility that there was a 27th resistance mechanism, and for which right now we don't know that there is an antibiotic.
Sure. So entirely I mean, if there's no way for me to test it, then we couldn't find it. So, but you're right. I mean, I don't know, you know, you can only study what you can measure. So I showed you one example with the bacitracin enzyme, which is a brand new mechanism. And then when, of course, when you look, it's not, you know, if someone would have cared enough to look in another organism, they probably would have found it, because we found it in surface organisms. There's nothing special about the cave, except that it helps isolate these organisms from the surface. But we did find, I mean, I didn't have time to tell you all the different other stories. I mean, I could give an hour talk just on that one strain of all the different mechanisms that we found, some of which were predicted. Like you could, you know, you see something that looks like it, purify it, sure enough, it does what that does. But then we found like that organism is not resistant to the natural product called erythromycin. It's a macrolide antibiotic, the first macrolide discovered, but it is resistant to a semi-synthetic derivative of erythromycin called telithromycin, which is a sort of ketolide antibiotic that was developed to overcome erythromycin resistance. So it's totally paradoxical. And we drilled down on that and did a bunch of studies. And I did have a bunch of slides in that, but I took them out because otherwise the talk would be an hour and a half, or you'd fall asleep. But there's lots of stuff to learn from these things. But of course, you have to have, now our panel of we just did a big study where we did 200 isolates, soil isolates from a single soil sample against 60 different antibiotics. And so the more you probe, the more you can find. And so now we're digging down hard on trying to find all these things. We've sequenced all 200, and we're trying. It's going to be a lot of work, but it's going to be interesting.
Yeah, all right. Thank you.
Okay. Do we have no more questions from folks?
No.
Great. In that case, thank you again very much, Gerry. That was excellent talk. Great way to end it off for the year.
Good. Good. I'm glad. And then, you know, I'm very hopeful that at some point I'll be out on a rock to come and actually visit in person. So I'm looking forward to it. So thank you very much again for the invitation, and you guys all have a fantastic holiday, and hopefully happy science. Okay, take care. Bye.